Hello everyone, I'm Yuki Hosono, researcher at Osaka University. I'm delighted to introduce our research, identification of alpha-galactosylceramide as an endogenous mammalian antigen for iNKT cells. Natural killer T cells are members of T cells which recognize glycolipid antigens presented by CD1D. iNKT cells support the induction of innate and adaptive immunity. Endogenous antigens for iNKT cells are necessary for development, maintenance, and activation. However, such antigens are not fully understood. Recently, several papers have suggested that alpha-GalCer might be present in mammals. However, the amount of endogenous antigens in the samples is very small and separation ability by chromatography is limited. So, the presence of such lipid has not been clearly proved. In our study, we identified alpha-GalCer in mammals for the first time using supercritical fluid chromatography. First, NKT hybridoma cells were cultured with those tumor cells without exogenous antigens. We found that CD1D transfected B16 cells activated NKT cells. We then performed transplantation tumor model. As shown here, tumor growth was suppressed in a CD1D dependent manner in vivo. We tried to identify antigen from tumor cells, however, no active fraction was detected. We tested the possibility that environmental ingredients contain antigen for NKT cells. We cultured B16 cells in 10% or low concentrations of FBS media. B16 cells cultured in low concentration of FBS media did not activate NKT cells. We confirmed that B16 cells cultured in lower FBS media activated NKT cells by adding synthesized alpha-GalCer. The results suggest that antigens on B16 cells were derived from serum. We purified the candidate antigen in the serum by liquid chromatography. We tested the similar activity using this self-assay system. And this fraction showed activity. From the result of TLC, this fraction was supposed to contain hexoceramides. Beta-GalCer or beta-glucel cannot be ligands for NKT cells. So, we saw that alpha-GalCer or alpha-glucel might be present in this fraction. To detect alpha-GalCer or alpha-glucel, we needed to separate these four diastereomers. So, we synthesized and prepared these 8 hexoceramides. We performed separation experiments using supercritical fluid chromatography, SFC. SFC uses supercritical CO2 as a mobile phase and provides improved chromatographic resolutions. We tested these 20 columns to evaluate the separation quality. Finally, we found that P4VP column provides the highest separation ability. As shown here, by using SFC, we succeeded in separating 4 diastereomers according to their retention time. Using SFC, we detected the peak coincident with synthesized alpha-GalCer, the 18-0-16-0 in serum. Mass spectrums showed that fragmented ions were identical with synthesized alpha-GalCer. Moreover, by adding synthesized alpha-GalCer to the serum sample, the intensity of precursor ion and product ions were increased. These results show that serum precisely contain alpha-GalCer. NKT cells are abundantly present in liver, so we analyzed bovine bile. We also detected several species of alpha-GalCer in bile. We also detect alpha-GalCer in marine lymph tissues such as spleen and thymus. Finally, we detected alpha-GalCers in human serum. This is summary. We detected alpha-GalCer in mammals. An environmental self-antigen can be utilized for antitumor immunity. Thank you for your attention.